Good morning, everybody. Welcome to Pediatric Grand Rounds. Thanks to those of you who are here in the room with us, and we have snacks here, but also thanks to you, those of you who are here with us online. We can see you there. Now I get to turn things over to Dr. Kapoor, who's going to introduce our lectureship and speaker for today. Thanks.
Thanks, Suzanne. It's a real privilege today to be able to host again Ronald J. Lemire Provider Grand Rounds. This is where we honor the legacy of Ron Lemire, who was a pivotal figure here at Seattle Children's for many decades and was part of that collection of luminaries, really, who developed dysmorphology and teratology as being centered in Seattle for so many years. And on the behalf of the Department of Laboratories, I have the honor of being able to invite a guest lecturer to present, and just thrilled to have Michael Wangler present today. Michael is a board-certified medical geneticist and human genetics researcher who's based in Texas at Baylor University. He's a professor of molecular and human genetics, and his clinical expertise includes paroxysmal disorders, boy, that's a mouthful, a rare group of pediatric brain disorders resulting from particular defect in body chemistry. He also specializes in disorders of intestinal hypomotility, which is how I got to know Michael, which represents a group of rare, difficult-to-treat diseases. He works on autism spectrum disorders, as well as a variety of other neurological disorders. Dr. Wangler is truly a physician-scientist, and he, I think, epitomizes sort of the paradigm of from patient to laboratory and back to patient again, physician-scientist model. And I think he's going to illustrate that very well with the talk you're about to hear. So, Michael, thank you for coming, and looking forward to hearing more about your work.
Well, thank you so much to Raj and all of you for inviting me here to Seattle Children's. And it's a real honor to give a lecture like this. So as Raj mentioned, I was trained as a pediatrician and medical geneticist, and I always sort of enjoyed the process of thinking about diagnosis. And I also have always been fascinated by genetics and fruit fly research. And so I'm going to talk to you today about kind of our journey in trying to solve a number of rare and undiagnosed disorders using model organisms. So I have no relevant financial disclosures for this lecture today. What I'm going to try to do today is give an overview of pursuing undiagnosed and rare disease diagnosis using model organisms, and in particular I'll focus on fruit fly models, which is what we use in our lab. I'm going to give you 3 specific case examples of patients and families that we've had who've been given a diagnosis through model organism studies and efforts. And then I'd like to give you some thoughts I have about sort of what is the justification for this kind of research into rare disease and using model systems for the wider benefit of medicine. So in our lab, we kind of have 2 main arms. We're engaged in trying to take patients who have an unknown kind of cause for their medical symptoms and use genomic technology to identify candidate genes that we study in Drosophila. And then we have a set of known Mendelian diseases, and we use genetic studies to try to understand the mechanism of those disorders. And I think the usefulness of something like Drosophila is not really debated in science, but where we've put a lot of effort is trying to integrate this type of research into the actual diagnosis of patients who are waiting on answers. And that's something that we talked about in this review article, really integrating work in non-mammalian models into the diagnosis of rare genetic diseases. In our lab, we like working with Drosophila or the fruit fly. Some of the reasons for this, they have a relatively short generation time, so it takes about 10 days between generations, and then they have a 70 to 80-day lifespan. They're excellent genetic tools that are often made in large collections across the genome and are freely available. And it's easy to culture flies in numbers. And then there's a number of detailed experiments that can range from behavioral observations, lifespan, all the way down to very intricate neurophysiology. We've been involved in particularly the fruit fly efforts for the Undiagnosed Diseases Network. Many of you are familiar. There may be people involved in the University of Washington and Seattle Center for the UDN here on the lecture call today, or here in the room. But for those who don't know, the UDN is an NIH-funded research network. It was an extension of the NIH Undiagnosed Disease Program. And it's essentially designed to use in-depth clinical expert evaluations, specialized testing, and genomic sequencing to find a diagnosis for undiagnosed cases. And usually these cases are selected for sort of having no clear diagnosis, but having objective findings and very unique clinical presentations that may suggest a genetic cause or a cause that can be identified through these investigations. The UDN has conducted over 3,400 evaluations across what are now up to over 20 sites. There's been over 2,500 exome or genomes for participants, and 940 participants have been diagnosed. And for the model organism efforts that we've done for almost a decade now, we've evaluated 526 total variants for whether those variants could be modeled to aid in the diagnosis. And there's been a number of manuscripts published from the UDN. So how does the model organism part work for the UDN? So basically, if patients are coming to the clinical sites and get evaluated and they undergo sequencing, and then a candidate gene is identified, that candidate gene can be submitted to the Model Organism Center. So what we do is we do a bioinformatics step first that goes from human down to the different model systems involved to try to understand both what's being proposed about the gene in humans and then what could possibly be done in the model system. Then cases are assigned to either fly, zebrafish, or worm, and then the research is done into the gene and the variant. That research is fed back to the clinical sites to help inform diagnosis. And then the clinical sites also provide a lot of clinical data just to help inform what kind of assays and experiments are done in the model. How do we select between the three model systems? Typically, there's one of them will emerge as having some advantage in the question that's being asked. For instance, it could be homology. There are some genes that have an ortholog in zebrafish, but they don't have orthologs in fly or worms. There could be where all three of the organisms, there could be genetic studies performed, but worms are perhaps much cheaper. Or it could be that fly has the tools that allow looking at the specific variant. So after selection, we've had a number of successes, and the thing I want to emphasize here is that in each of these stories, there were sort of a patient that started out without a diagnosis, and at the end, there was a specific genetic cause attributed to their clinical disorder. So we grouped these into what we call gene discovery. That's where a gene was not previously associated with human disease. And then we also have phenotypic expansion, where we were familiar with the gene in medical genetics, but the patient's clinical phenotype differed from what we were familiar with, and we were able to do some experiments that showed that that variant has a different activity or a different inheritance. So in order to use this type of research with patients that are waiting on a diagnosis, who are seeing clinicians across the network, we've had to do several things. So one of the main things is the monthly group calls. So we basically have a working group, and the format of these calls is typically we'll start out with the clinician who will discuss the clinical findings in the patient, and the clinician will discuss why they felt that this candidate gene was of interest, and then it'll move into each organism, what each organism has in the equivalent gene, and then we'll sort of come together with a plan for the patient where the research could inform the diagnosis. This is very collaborative, and we sort of all work together. It also is very dependent on connecting with other patients around the world. And for most of these projects, N of 1 is still not enough in human to draw clear conclusions. So we put a lot of effort into things like matchmaking through Gene Matcher, or there's other genomic resources like the Greger Consortium to try to see, are there other patients with overlapping phenotypes. One thing that we discuss is that the first step in the model organism is the loss of function phenotype. So we could have cases where there's a very interesting variant, and we think that variant has very interesting activity, but we really need to start with what is the loss of function phenotype in the model. But then we do move on to variant-specific studies where we take the patient's variant and study it in the model. And for those, we usually are also kind of on a time clock because we want to return some meaningful result to the network. And so for worm and fly studies, this typically is a 6-month clock. And then for fish studies, this is a 12-month clock. This doesn't mean we have everything figured out at the end of 6 months, but usually we at least know whether there's something of interest that we could continue to explore. So how could we do this sort of last part of variant-specific studies? In fly, this maybe look like a complex slide, but let me kind of explain it. So we use a process that we call humanization. So if we have a gene of interest in humans for a potential undiagnosed patient, we can go to the fly ortholog, and the key is to integrate this cassette that we call the T2A into the intron of that gene in the fly. And what this allows is it actually disrupts the fly gene. So it makes a loss of function for the fly gene, but it produces this GAL4 transactivator protein from those same enhancers. So within the same cells, this will be produced, and this can then be used to drive something like a fluorescent marker if we want to see where is it expressed, but we can also drive the human gene itself. And so then this would allow us to compare what we would call the reference sequence to the patient's variant. And so this is our preferred strategy. And for us, about 60% of fly genes that are, from this project can be successfully humanized using this method. So I'm going to now turn to the sort of 3 case examples. So the first one, I'll tell you a couple stories. So at the top was a patient named Erin, and she was born with muscle tone issues, had a developmental delay, and a lot of tremors and muscle weakness over a long period of time. And she was enrolled at the Harvard site for the Undiagnosed Diseases Network. And around the same time, a patient of ours at Baylor named Oliver, who had some issues with missing developmental milestones and had been diagnosed with cerebral palsy, sort of came to our attention. And around the same time, we actually found that both Aaron and Oliver had an identical de novo variant in a gene called DNM1L. So part of our lab is very interested in organelles, peroxisomes, and in particular, and we knew about DNM1L, and we had studied patients with DNM1L variants before. DNM1L is involved in this organelle fission event where peroxisomes can actually divide. And what was interesting about these two patients is they didn't fit what we already knew about this gene. So we knew there are patients that have inherited cases of optic atrophy over here in the GTPase domain. We knew there are patients with severe infantile epileptic encephalopathies that we had seen with variants in the middle domain, but they sort of had this unique variant out in the GED domain. So a postdoc in our lab obtained cells from one of the patients, and he was actually able to show that the peroxisomes in these cells showed this sort of tubulated feature. So you can see if you compare the control at the bottom. Oh, and actually I apologize to those online. I didn't follow the instructions. So now this maybe should show that the control sort of has the normal appearance of peroxisomes, and the patients have these tubulated peroxisomes. So this seemed to establish that this gene is indeed involved in their phenotype. It's just that their phenotype differs quite a bit from what's known. So then we went to the fruit fly, and here what we did is within the fruit fly, there's an organ called the salivary gland. And what we were able to do is compare a spectrum of different human variants in this gene across in terms of their impact on peroxisomes. So if we compare sort of the reference to the optic atrophy variant, we don't see much of a phenotype at all. The variants that are involved in early infantile epilepsy seem to show very severe effects on peroxisomes. But what's interesting is the patient, for our new patients, they sort of have an intermediate impact. So here, the fly was helpful in kind of telling us that this variant is likely in the same category as what we know are disease-causing variants, but the impact is different, probably due to the severity. So we actually confirmed this in additional tissues. This is actually showing muscle and looking at the peroxisomes in muscle. And again, we can kind of see that there's pretty severe effects of these epilepsy-causing variants, and our patient's variant are in an intermediate category. So here, this kind of has helped us think about that this new category of variable dominant negative activity is associated with patients with neuropathy, dystonia. It's actually potentially a consideration in patients that are suspected to have cerebral palsy, and that is very different from what we'd seen previously for this gene. And this has been an interesting project because it started out with the patients sort of applying to the Undiagnosed Disease Network, and now we kind of have this partnership where the patients have come to our institute. We've shown them flies that carry some of the same variants. They're working very hard with this foundation to support new research and new therapy into these disorders. So the second example I'm going to give is a different disorder, different gene. So also from the Undiagnosed Disease Network, and this has really been run by a postdoc in our lab named Jonathan Andrews. So for this case, we actually had two individuals, age 12 and 11, with intellectual disability, speech apraxia, and some dysmorphic features. Both patients had actually independently enrolled in the UDN. They didn't know about each other. One was at NIH site and one was at UCLA. They both had negative prior sequencing. And on research reanalysis, both individuals were found to have de novo missense variants in a gene called MRTFB. So Jonathan looked at this And he found this to be kind of interesting because MRTFB is a transcription regulator, transcription factor, and it's involved in the equilibrium of actin within the cell. So in the presence of a lot of G-actin, the MRTFB is bound and remains out in the cytoplasm. But then when there's a conversion to F-actin, more MRTFB can go into the nucleus and activate transcription. And it does this with a binding partner called SRF. And Jonathan noticed that actually one of these patients has an arginine substitution right in the binding domain for actin, for this protein. So basically, he noticed that this substitution is highly conserved, and predicted that this could affect the binding of this factor. So what he did is he expressed these in the wing of the fly, and these are fly wings. So you can see the pattern of a normal fly wing, and what you can see is that the human reference for this gene doesn't have much of an impact, but the variants definitely have a strong impact. And Jonathan went on to look at, based on our knowledge of how this works, he brought in the SRF, the binding partner, and he actually saw that induced an even stronger phenotype in the presence of the patient variants. So he developed this hypothesis that the patient variants are essentially gain of function. So it means that, the patient's version of MRTFB is not able to bind the G-actin and is kind of inappropriately going and impacting expression. And so then he thought, I wonder if their cells have some changes in actin itself that we could see. And so this experiment, in this experiment, we actually have the green cells within the fly ovary mark where the mutation is. And what we can see is that in the green cells, actin appears very irregular. It's sort of aggregated in these cells very abnormally. So we thought this was exciting because it sort of suggests ways we could think about treating this disorder, because you could imagine, could we prevent the interaction between the two partners? Could we prevent the MRTFB from entering the nucleus? But really the most interesting one would be, could we just remove this sort of gain of function? And of course, now the technology is increasingly becoming available to treat patients with antisense oligos. And so what Jonathan is doing now is he's designed a series of these oligos. But what was key is that we have a phenotype in the patient cells related to actin that we can use to screen the effectiveness of these treatments. And what was nice is we wouldn't have gotten that far if we didn't have the fly model, because we were able to work out that actin would be aggregated in these cells. So I'm going to move on to a third example. And so our work in the UDN sort of led us to think about other ways to do similar types of projects. And one of the projects that we've been really excited about is called the Community Texome Project. And this really arose from within our hospital. There were patients that we were not able to enroll in the UDN. It could be transportation issues. It could be that the workup is not complete. And so we focused on sort of healthcare, underserved populations, under-resourced. And the idea behind the Texome is that we performed the sequencing at Baylor. And then another highlight that I'm going to go to briefly here is the role of machine learning in genomics. And then we sort of have highlighted that tool as something that we're testing. And then moving on to the fly studies. And in this study, we provide longitudinal follow-up, so we continue to try to find new genes for unsolved cases, and all the patients receive genetic counseling. So our Texome project is toward the end of its 4th year. You can see we actually have a mixture of adult and pediatric cases, and our diagnostic yield in the project is about 28.6%. That includes cases where the sequencing has solved the case, and then also we include partially or probably solved where there's a likely diagnosis that requires more confirmation. So what about the unsolved cases? So for Texome, we sort of don't have an entire team like the UDN to help select candidate variants. So we tried to think with an informatics team at our institute of a creative way to try to use AI or machine learning to suggest candidate genes. So basically, if a patient undergoes sequencing and they remain unsolved, first we reflex to trio if we hadn't done that already. If there are any research candidates we can find, we can pursue those in the model. But then we turn to this AI Marvel tool, which was developed by our team. And basically the AI Marvel takes into account a number of factors that I'll show to try to select candidate genes and point them out to you. And then, at least for us, this process is great at coming up with new ideas, but all the new ideas that it comes up with, we have to manually go through and select. Some of them just don't make sense. But so we rely on sort of validation. We submit the strongest candidates to GeneMatcher to try to find other potential patients, and then we do fly modeling. So the AI Marvel tool, it basically takes into account the phenotype. It has a way that we can input the clinical phenotype into, we use HPO terms, which are phenotype ontology terms. It will annotate the phenotype. Then it identifies features. By feature engineering, what we mean is that it's looking at features of the case and features of the submitted gene, like where it's expressed, in order to try to kind of come up with, does this work based on its prior experience? And then it gives an AI-based scoring system. So it literally just outputs a list of, number 1, number 2, number 3, number 4. And then we go through that list in some detail to try to see, do we think this makes sense? So this is just showing that it's what types of information it takes into account at each phase. And the goal is really to identify candidate genes that we will then validate or model. So for the AI Marvel, just to give you, I want to give you a clear perspective on how, how this is being used. So we've basically had about half of our research candidates have come from just that reflex sequencing or the manual process that we normally do. But for 27% of the cases, we do have a candidate that the machine learning identified that we didn't initially come up with. And then 44% of the cases, we still don't have a good candidate. So this is a list of some of the genes that have been identified by the AI Marvel. And we do have one that has become a success story. So this gene is called BRSK1. So within our Texome project, we had a 4-year-old male with global developmental delay, short stature, and hypotonia. That we identified this missense variant in this BRSK1 gene. This is a brain-related kinase. The variant's not present in the NOMAD data. And so, and I should mention that this was not the top AI selected. This was actually in second place initially. So we pursued two candidates from the list, but this one had hits from GeneMatcher where we thought, hey, it looks like there's patients with overlapping phenotypes, and they have variants in the same gene. And so then we've been pursuing, again, Drosophila work to try to look at some of these variants. So this is showing the homology. The Drosophila gene was already known. It's called the SFF gene. And you can see that there's conservation of several of the patient variants, although some of them are also stop gains, so meaning loss of function mutations. And so we pursued the same approach that I mentioned, the humanization, where we are comparing the reference and variant sequences. And here what we did is we took the flies and put them into a climbing assay. So we're testing out the climbing ability. And what you can see is that in the control, they're able to climb sort of after being tapped to the bottom of the vial. When they're missing SFF, they're really bad at climbing. They're very weak. The human gene can rescue that ability in the fly, but the variants don't rescue it very well. So this alone, this assay alone, I don't think we'd consider this like enough evidence, but it's interesting. There was recently a paper by another group showing neurodevelopmental and particularly epilepsy phenotypes for BRSK1. And this is interesting because only one of our patients has seizures, but we took this as additional evidence that we're onto something with this candidate. Usually it's too bad when another group publishes the gene first, but here, since we're working kind of on this premise of AI helping us select, we actually felt good, when we sort of saw that this is being validated by another group. So I guess I'm going to take a few more minutes and try to give you, these are more like big thoughts about the importance of this type of work. So, why is it worthwhile to do this type of work? So because there's several arguments that one could think about. These are very rare conditions, and so it's a small number of patients is one argument. And then there's also been, more discussion about what is the role of model organism research more broadly. But I really feel strongly that this work is important to do. So the first point I'd like to make is that this ends the diagnostic odyssey of the patient. So they don't continue to pursue additional tests in many cases. They're able to kind of center on a clear molecular diagnosis rather than things that maybe sort of seem more vague. It also avoids the diagnostic odyssey for future patients, because for each of these genes, diagnostic labs then adopt the gene, and some of the genes from the early part of the UDN model system work, we now have more than 100 cases that we're aware of that have been diagnosed based on that gene. And then I think for us, a case like the MRTFB, where we're trying to go towards a therapy, is important. If we do the model system work, we often have an idea about what we're trying to do with therapy that we might not have if it just started out as sequencing. And then I'll kind of go into the hardest one, which is, can this provide insight into disease mechanism for common disease? So, I definitely have had a lot of, it's been an honor to meet a lot of patients who have sort of had an end to their diagnostic odyssey. And this has included patients like Avery, who she was diagnosed through UDN efforts, but part of the way I met Raj was related to actin gamma 2, and patients like JW sort of had sequencing fairly early in infancy and identified the gene fairly early. And then this is Kai, who also was diagnosed sort of after other patients were found in the UDN with this mutation. And in all these cases, the families have started very productive family foundations and have really tried to advance research studies into the gene. So that's kind of my argument about the diagnostic odyssey. The the sort of third argument was guiding therapy. And, I think the point I want to make here is that when we're doing this type of work, we identify the variants that are disease-causing, but we also have some idea about what is the issue, what's the molecular mechanism. Is it a loss of function where we would need to somehow replace the function of the gene? Is it a gain of function where that allele, that mutation is toxic? And that can really lead us as we start thinking about mechanisms and therapies. Are we looking for something like developing gene therapy or antisense oligos? Maybe it helps us with drug screens to know which target are we screening and which phenotype we're screening, and that can be started in the model systems that we develop. And then, the hope is that this leads to personalized therapies through gene therapy or gene editing. Maybe it allows us to target specific pathways. And then also, we still feel like looking at FDA-approved drug repurposing is also a very productive approach that we can start in the models. But then the last one that I think is the hardest is, sort of arguing how do we take this type of work to help all kinds of diseases and, common disorders as well. And I sort of want to share with you three examples that I've been thinking about that I think the argument here is like we should be patient. Because if we look back over several decades, there definitely are examples, and to me they fall into different paradigms. So I guess I would put forward the RAS pathway. So this is a pathway that really, you can't argue that this was sort of found through rare diseases. This was a well-established pathway in cancer, starting out with studies of viral-induced cancers. But rare disease mutations from conditions like Costello and cardiofaciocutaneous syndrome have been a help in guiding cancer therapy towards like less toxicity, right? So the rare disease mutations help us see these are sort of allowable changes in these genes and these are not allowable changes. So that's something where the rare disease variants can help us inform a well-established pathway. And many of the genes we have, we can start looking at where do they fit into something that's already well known. Then there's, the example for me, of Alzheimer's disease, that the rare early onset forms of AD really did help revolutionize AD research. So I think the specific therapeutic targeting of things like amyloid, maybe that's remaining to be seen, whether that will be successful. But a lot of the patients who had the rare early onset forms, they definitely brought a molecular understanding of AD plaques, and that really helped that field become much more molecularly oriented. And then the final is like the best case, something like PCSK9, where you have a rare disease in a cholesterol metabolism pathway, and that becomes a direct therapeutic target. And so we're definitely also looking for those. So I'm going to just circle back briefly to our work on DNM1L. So we actually have been interested in the role of Alzheimer's-related proteins and how they impact peroxisomes, because we have so much interest in peroxisomal disorders. And so what Saurabh has been doing is he's actually looked at how the expression of tau, Alzheimer's-related protein, can really alter the distribution of peroxisomes. And but what's interesting is that we sort of see effects the other direction as well, where we bring in some of the disease-causing mutations in DNM1L gene, the gene I introduced in the beginning, and we can see that that actually can remove tau or suppress the expression of tau in fly brain. And so we're looking at this now in the patient cells as well. And so, I'm hoping for us, one example that we'll develop is this connection to Alzheimer's disease, because when we have explored rare peroxisomal disorders, we've actually started to notice, and it's not just us, there's other people in the field that also notice connections to sort of the biochemical features that we see in AD brain. And then, DNM1L actually interacts with tau and APP. So this can all link organelle dynamics to a common disorder and dementia. And the point here is that we learned a lot from the rare mutations that gave us a lot of tools that we can then try to translate into things that we observe in the common disorders. So hopefully I convinced you that model organisms can actually aid in genetic disease discovery, in diagnosis, and that the genetic mechanisms are important to guide personalized therapy. And then also just that these efforts involving like patients, clinicians, researchers, all working together to treat rare disease really, could help us move into a era of personalized medicine. And just want to thank, I'm just going to show this. This is our lab, and we're located in Houston at the Duncan Neurological Research Institute. And then, more broadly, this is a really big team. A lot of the data I've shown today involved efforts of Hugo Bellin's lab, Shinya Yamamoto's lab, participation of patients and families, and then the UDN clinical sites and sequencing teams. So this is definitely team research. So I think with that, I'm going to put this thing, and I can take questions.
Thank you. If you're in the room and you have a question, raise your hand so I can see it.
Wonderful talk, Michael. Thank you so much.
Thank you.
That AI Marvel tool that you talked about, is that available to people outside your laboratory or institution?
Yes.
And if you would sort of dovetail that with, you mentioned, I think, with the patients who had the MRTF, the index patients, that next generation sequencing initially didn't find any variants. How often are you seeing that? And is that AI Marvel tool something that we all should be using when we get a negative result to reanalyze somehow the... And
Yeah.
why are we getting these negative results?
No, good question. Because, so I think basically the workflow for both studies is really that the patient undergoes sequencing and they get a clinical report. It's research, of course, but they get a clinical report. The clinical report will focus on known established genes. It'll use the ACMG variant classification to tell you it's likely pathogenic, likely benign. But then from there, each of the studies sort of has this workflow of where we would then take the raw data and look deeper, particularly in the unsolved. And AI Marvel is available to anyone, so it's available on GitHub, for instance, for people to set up on their own. It's also available online. I do just try to explain, though, that in our hands, these are patients that we're looking for new ideas. We don't have any gene candidates. Some of the candidates that AI Marvel comes up with, you can immediately say, for instance, we had a case where tuberous sclerosis was the top a variant in the TSC1 was the top candidate. And we knew the patient didn't have this condition. But you sort of look back and realize that somewhere in the notes it talked about hypopigmented spots, and you sort of see where it's coming up with some of these ideas. So for us, the point is to use it to come up with candidates that we look at deeper, and it sort of takes a manual process from there. John, yeah.
It was an interesting talk, Michael. Thank you.
you.
For those of us who work in model organisms, one of the common criticisms that we get is, and in your case, you're putting a single gene into the network of a different organism
Right.
with different orthology
Right.
and all the rest of it.
Right.
And your example with MRTFB and SRF is a good, I think, illustrates
Right.
part of the problem. Maybe you could just comment on it, because you had to put SRF back in
Right.
to reveal the phenotype. And so you had the network information and you had to put the human gene back in. Maybe
Right.
just comment on what sort of problems you faced
Yes.
and how you've overcome them, and what the community thinks about this sort of approach.
Yeah, no, great question. So I think the network effects often come into play in that, like one example is human genes can sometimes appear very toxic when we express them in the fly. And a lot of those we just sort of note anecdotally that they're part of some complex, or have important binding partners. And in those, we maybe don't always go to the trouble of proving it, but the idea is that it maybe doesn't interact with the binding partner in the fly the way that it does in human. And that kind of explains some of the toxicity. And it sort of depends if we're just kind of grinding down to we want to just prove that this variant is different. We want some evidence that it differs in a meaningful way. We may sort of continue knowing that there are those potential complications. But then in other cases, we may decide, okay, now we need to compare human SRF and human MRTFB. Or I would say the most practical approach would be testing in the fly. So if it's a conserved site, testing in the fly context helps us a lot, because we've had cases where the human expression of the human may as well be a sledgehammer or something, but then you can make the variant in the fly transgene and you just see subtle differences. So I think it's very important to think about, and just sort of trying to use caution with all the interpretation.
We have one person online who I believe can unmute and ask his question, Brock Grill.
Yeah, I'm here. Hi, Michael. I'm Brock Grill.
Hi.
I couldn't make it down to the clinical site today. My research lab is downtown at the Research Institute. Couple interesting things. Love Aitchison's question, right? Spot on. Actually, my lab study identifying a novel neurodevelopmental disorder using gene editing, CRISPR gene editing in C. elegans, is one of the gene diseases that came out of Marvel's study, your paper, together with Hugo and others.
Okay.
It's one of your proof of principle candidates. So I was like, I'm a huge proponent of this. I
great.
think I loved how you pitched a case for invertebrate models. I think the point about subtle phenotypes is really well taken and highly valuable. So the interesting thing that crosses my mind is transgenic rescue versus direct CRISPR gene editing into the native locus. What's your thoughts on this? Have you guys tried both? Do you see differences? What do you think?
Sure. We haven't done as much of the direct gene editing approach. And I do understand the question, because that does seem intuitively simpler. I guess one thing is for instance, the T2A tool that I mentioned, we get a lot more out of that. So for instance, it's so versatile, so we can use it to cross to a fluorescent marker and see what cells express the gene. We can cross it to both human and fly transgene versions. So I think the versatility has made that our favorite approach. We also do sometimes engineer the change in the fly genome, but maybe that's even in a genomic construct. So I think engineering it in the genome is also definitely valid. It just, in our hands, for us, we more typically wanted the versatility of these tools. They're also, the other point to make though is they're also available in collections usually. So a lot of times we're just ordering lines that have already been established. Good question though, thank you.
Thanks for this
Thank
informative
you.
talk. I'm an immunologist and I'm taking care of primary immune deficiency diseases. And there are about 500 or more genes that are involved in the immune
Right.
system.
Right.
Now, how do you assess the immune system in a fly?
Right.
I mean, do they
Yeah.
have a thymus
Yeah.
equivalent?
I mean,
Do
I
they
will.
have
No, yeah.
hematopoietic stem cells?
Yeah, yeah. No, I will say that so two answers. The first answer is if it's below adaptive immunity, so toll-like receptor, the natural killer level immunity is actually pretty well conserved in the fly, and there can be some direct assays. There's something called crystal cells that can be assessed in the fly, and there's pigmentation phenotypes that can result from these. But for us, like things in adaptive immunity, probably a direct one-on-one comparison isn't possible. I will point out though, like we sometimes will still use the fly model for kind of a focused question. So if it's a conserved gene and we just want to compare the mutation and the reference sequence, we could take it into the wing and the phenotype might look nothing like what the patient's phenotype is, but we still can see molecular mechanisms. That's the key, is we could distinguish gain from loss of function, and that could still be valuable information even though the phenotype is completely different.
Do they have stats, for instance, stat one?
Yes, they do, yes.
Which can
Yes.
gain of function or loss of function?
Yes.
patients?
Yes, definitely. I think in that case, though, it's a case where there's essentially one gene in the fly genome that represents the vertebrate ortholog, like several vertebrate orthologs. But yeah.
Great talk, really interesting. My research usually starts with mice, and I noticed you didn't use the word mouse a single
Once.
time.
I'm
So
sorry.
I'm curious
I'm
about.
sorry, yes.
No, it was very refreshing. I'm curious about, like, for the things that were
Yes.
the best hits, do they continue on into mice, or is
Yes.
the phenotype too subtle to see in mice, or does it just take too long?
No, great. Thank you for the question, because it was an omission. I should. So we actually we do have at Baylor a team involved in it's called Centers for Precision Medicine, and the idea there is to make knock-in mice for specific variants, particularly to answer therapeutic questions. And a lot of these cases from the UDN where we maybe started and found some promise, we would then submit for mice. And I think I should not have neglected that, because that's obviously a crucial organism to go to sort of after this work is established. Yeah.
Hi, thank you for the very interesting talk. My question is related to the first part that was about DNM1L and expanding the phenotype of the gene.
Yeah.
Could you speak more to the genotype-phenotype correlation and some of the challenges that you had, because you were, maybe I missed that in the, that image from the gene domains.
Yes.
Was this the first and only variant that you found in the last domain of the gene that was related to cerebral palsy and none of the, not epilepsy?
We have focused on a missense variant in the GTPase domain that is associated with dominant inherited optic atrophy. That's essentially over, they have the mutation for several decades, and then there's a slow optic atrophy phenotype. There are other variants in this domain that are associated with like a recessive form of a sort of loss of DNM1L that I didn't mention. But the middle domain had been that's been where most of the attention has been, because a lot of these variants are de novo. They're often found in infants with epileptic encephalopathy, and there can be brain MRI changes. And what was unique is, our patients over here, they really don't have a lot of cognitive impact, and they don't have seizures. So they have a very different phenotype that's essentially a motor phenotype. And we weren't sure what we would find when comparing these. And it seems like what characterizes them is that those variants are kind of in between the mild and severe impact. But we did have to do many different assays to see that, because some assays, it may look as severe as the severe cases, and other assays, it might look more mild. So I'm not sure if that answers your question, but when we first saw the patient, it wasn't obvious that they could have the same gene involved.
Thank you.
Yeah.
Anybody else? All right. Oh, yeah. Last question. Thanks.
So a lot of us encounter patients with undiagnosed disorders. How do we access, or can any of us access the UDN? And do we have to have 2 patients? And are you flooded with patients, or are you looking for them?
No, I think so. I mean, for the UDN, the process of application is actually through the online. There's an online gateway, and it's a family-driven process. So if the family wants to, they are the ones that essentially apply. But then they do have to. There does have to be a letter from a clinician, essentially providing some justification. The main things that are being looked at are their objective findings. So something on a EEG or brain MRI or an assessment by a rheumatologist or a neurologist showing, objective changes, and then the uniqueness of the clinical presentation are going to factor into their decision. I imagine if they're here in town, they would be assigned for evaluation to the, I think Dr. Jarvik calls it the Pacific Northwest UDN site, but it's, they'd be assigned here. But I think there's also other options, coming up. A lot of centers are opening undiagnosed disease programs. So I think, and then even just researchers with specific interest doing exome and genome reanalysis and trying to develop, those kind of programs. So yeah.